Today we will discuss about another paper where they have used a knockout library of murine genes. Around 915 murine genes they have used in order to create the CRISPR knockout library, and then check those knockout libraries, check within those knockout libraries about the cells that will give them a highest migration. So this is in the panel A. It has they have shown their brief overview of the experiment what they have did. So first they produced this knockout library using the lentivirus guide RNA. They prepared the uh, plasmids that contains 5 guide RNAs for each gene. After they have did the transduction of those knockout libraries into these uh, pancreatic cells, and thereafter they did the migration screenings, and in these migration screenings they have separated out the cells that gives them highest migrations after 6 hours in the transwell assay, and did the genomic DNA isolations, and thereafter they did the NGS sequencing to get an idea about the genes that gives them the highest migration. And when they did the analysis among their knockout libraries, which genes gives them the highest migration, they are able to find out that STK11 gene gives them the highest migrations. And however, when they compare all the genes in terms of that which of these genes will give them accelerated migrations, they're able to see that around 125 genes, that is they have considered from this number of good sgRNAs per gene, from around 135 among around 135 genes has a good sgRNA representations in the accelerated migrations, that has either one good sgRNAs or more than one good sgRNAs. And one more thing that they have found out, that out of the 5 guide RNAs that they have used for the STK11, 3 guide RNAs are uh, 3 guide RNAs are expressed in this accelerated migrated cells, or 3 good sgRNAs of the STK11s are been observed in those accelerated migrated cells. And when they check the read counts of all the STK sgRNA genes, they are able to find out that this STK11 sgRNAs are expressed in 3 STK sgRNAs are expressed or has a higher read counts compared to the other case, compared to the this control case, or compared to the other 2 guide RNAs that are shown in the treatment. After validating that it's the STK11 genes that has the highest migratory among the murine knockout libraries, murine genes knockout 911 murine gene knockout libraries that they have prepared. So they decided to again confirm this STK11 in different cell lines before moving further into the experiments that are based on STK11. So what they did is they checked the STK11 presence in different cell lines that they have in their lab, and they are able to see that for this TKCC10 and Mayo 4636, the STK11's presence is negligible, whereas for the PANC1 cells, which is the pancreatic cancer cells, has the more STK11 formations. So this indicates that, and also this PANC0504 also has the STK11's presence. So this indicates that the STK11 could be present in the pancreatic cancer cells, that is pancreatic cells that have acquired the cancer properties, and uh, contribute this could contribute the pancreatic cancer cells towards its migration. And for the control they have used this GAPDH, and from their preliminary analysis, they seems to be more focused on the STK11 for their further experiments, and we will discuss their subsequent experiments in our next videos. And before ending this video, I would like to say that whatever the finding they have find out is in line with the some of the clinical treatments that are done based on their findings, such as this STK. Some of the therapeutic treatments that are available target STK11 genes to prevent the migrations of the pancreatic cancer cells, and it has also leads to the increase in the survival rate of the patients who are affected with pancreatic cancers. So we will discuss all these things in our subsequent videos, and if you need to understand or if you need to read all these things in advance so that you can understand our subsequent videos in a better manner, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.